either go to left or go to right. And if it chooses left, then it will be rewarded, okay, if the order was cheese, and otherwise it would be rewarded if the order is chocolate. So the rat is learning a task to recognize the order and make the right decision to go left or right. Now, during this task, the activity is recorded, okay, here in the medial prefrontal cortex, through a set of electrodes. So you have different electrodes, 8 of them, and these electrodes actually measure the activity of some cells. So the number of cells is about 100 cells, and this is typically a bit less than 1 million spikes.
Testing.
And if you see, you want me to?
Yeah, just for the recording.
Okay. Thank you. Okay, and again, if you look at the cross correlograms here for a pair of cells, for instance, then you may find a peak, positive peak or a negative peak depending on the pairs of cells. And then that helps you to define some interaction matrix, if you want, or correlation matrix more. But just a matrix of correlation indices I mentioned before. Okay? So you can get positive or negative interactions. And again, the distinction between interaction and correlation is not clear. So it's a very useful measure of, let's say, the connectivity map between the cells. And you can do much more than that. And actually, these people looked at how this connectivity map evolves with time and how it changes when the rat is moving through the maze. And some of the connections that you find actually do not depend on the location of the rat in the maze, but other ones actually change with time and depends on the location. Okay, so the purpose of all this analysis based on correlation indices is to get an idea of the connectivity map between the cells.
So is this an effective connectivity, or is it real? Because
it's an effective connectivity. That's what you get, let's say, functional connectivity. I don't know, effective, functional, whatever you want. I mean, it's not physiological here, okay? Now, you would expect that it's related to physiological connectivity, but the connection, I mean, the relation between the two is something which is not obvious at all, okay? It's what you get from the activity.
Sorry, what's the red and black arrows?
Sorry?
What do the red and black arrows?
Oh, sorry. So the black ones mean the ones that do not change with location, and the red ones are the ones which appear at some position in the maze. Here, it's a normalized position in the maze. So you can define a coordinate here, which goes from 0 to 1 along the path. Okay, and the cross correlogram for one particular pair of cell will be either flat, and it may change at some point. So you say that, okay, you have no connection here, then you get a connection, no connection anymore. In that case, you would get a red arrow saying that you have some time-dependent connectivity, if you want.
And this is, so neurons, they're recorded from, what kind of neurons are they? Are they head direction cells or something like that?
So you have pyramidal neurons and interneurons in the prefrontal cortex. I don't know if that answers the question.
Well,
I mean,
so because if they were recorded, for example, from the hippocampus, from some of the
yes.
cells,
you would have place cells or whatever.
place
Yes.
cells. So, for
Yes.
example, I was wondering whether you have this change in effective connectivity at that particular place in the maze, because the cell you recorded from actually as a place field for that.
Yes, so no, it's a good question. You know, I mean, I just inserted a figure from the paper, telling you what people would like to do. The interpretation, I mean, you have cross-correlogram, and you see how they obtain that. You have change in the cross-correlogram as the rat is moving. Now, whether this should be interpreted as a change in the connectivity is something we are not, I mean, forced to agree with that, okay? It could be simply a change in the activity and the connectivity remains the same. So, but basically, I just want to say that people use cross-correlogram to establish connectivity maps. So it's a well-known fact, and that has been going on for decades. So, but it's well known also that there are problems with this kind of approach. And one of the problems is that if you detect some correlation, as I said before, that doesn't mean that there is an interaction. Okay? It might be that you get interactions, or let's say, between two different cells. Here, I mean, you may get a correlation between these two cells here simply because they are interacting through some other cell, for instance. Okay, so you may have correlations which are not direct correlation, but just mediated correlations.
But you can probably tell that the time of the correlation is different because
Yeah, so that's one way to do that. So people actually, there is a very detailed analysis by Nicolas Brennan and collaborators 2 years ago about actually, depending on the time and if you can actually deduce whether it's a direct correlation or not, and it's not obvious at all. I mean, it depends very much on the details of the model. So,
And if there were some studies which actually studied real connectivity, like the slope of how do those correlate with this sort of testing?
you mean, so is the interaction we can get, are they related to the real physiological?
Have they been compared? Has this cross-correlation analysis been compared to the actual physiological, where you see the connections changing in real time?
Okay, so in that case, in the experiment I mentioned here, I don't think that has been done. Okay, so I don't want to insist too much on that. That's in their paper. Okay, I'm not going to say anything about that. I think it's a very speculative idea here. Okay, because you will need also a change in interaction on a very short time scale, which is something which why not? But I mean, it's very speculative. I think.
How does that time scale compare to what's with us now?
I don't know. I mean, we can go back to the paper. So, but what I want to tell you here is, let's say, I want to present some technique which is part of an effort to try to disentangle this kind of direct correlation from an indirect correlation. Okay. So basically what we want to do is, suppose you are given a set of spiking times here, so which is not the raw data. I mean, you have already, the first step will be spike sorting, okay? Suppose spiking is done. Now, and so you get this data, and you want to know, or you want to infer some connectivity map between the cells. Okay, so you want to know between cell I and J, there is a connection from cell I to J or J to I, also some external input currents and so on. Okay, so this is not new. I mean, this question is not new, and there have been many different approaches. So the one I want to tell you about here is something we have been doing, and other people have been doing so, is based on the integrate and fire model. So integrate and fire model of neurons are simple enough to be amenable to some mathematical treatment, and still they retain some, let's say, very basic features of real neurons, like the idea that there is a membrane potential, threshold crossing, and so on, and some very basic features. But I'm not claiming that they are realistic neurons, and they're highly simplified version of neurons. And we do that in the Bayesian approach. So I talk a little bit about other models we do as maximal entropy models, but I think we'll have a discussion tomorrow about that. And also generalized linear models. But most of the talk will be about integral and fire model. Okay, so in the Bayesian framework, we assume a model to generate the data from an interaction network. So the model, and I will just present it in the next slide, tells you that if you are given a set of interactions, for instance, then the data, which is a raster plot, for instance, will have some probability. Okay? And that's a model. So it's a stochastic model to generate activity given the interactions. And all the information about this distribution over the data is in this likelihood function here. Okay? So, and this is true in all models I was mentioning before in the previous slide. So I will tell you how this likelihood actually is defined in the next slide. Now, then we may have some prior information about the interaction, information which is not contained in the data, but we know, for instance, that some interaction should be positive, for instance, this statutory interaction. Some other one should be negative or whatever. I mean, so we have some prior information in some cases. We may impose, for instance, that most of the interaction should be equal to 0, or very close to 0. So we have a sparse network of interaction. So this kind of prior information may be very useful if the number of data we have is very small, okay, compared to it. You know, here we are looking at n cells, so n of the order of few tens, even few hundreds. And we want to infer a matrix of connectivity between hundreds of cells. You know? So it's a huge number of parameters. So we need a huge number of data to do that. If we don't have this data, then some prior information has to be put inside the model. Otherwise, that's just impossible. Whether the prior information is good or not, that's another question. And basically, what we want to do using Bayes' rule is maximize the posterior probability of the data, of the interaction, sorry, which is maximize the product of the likelihood of the data given the interaction times the prior information. and we want to maximize that over the interaction. Okay, so we are given the data and we want to maximize. So in the following, I will just forget about the prior, but I will talk about it just at the end. So the model we are considering is a leaky integrated fine model, stochastic version. So the equation here is the equation for the membrane, for the dynamical evolution of the membrane potential. So each neuron is characterized by a single real variable, which is the membrane potential, V, and the model is the following. So the change, let's say some current is entering the cell. So we have a current coming from other cells here. So each time a cell, so suppose this is your one cell here. And this is a pattern of activity. Actually, you have two patterns of activities here, the black one and the gray one. Okay? So consider only the black one first. So at the initial time, the potential of a neuron will be, for instance, equal to some rest value here. Now, the first spike here which enters, comes from input cell number 8, will produce an increase in the potential, for instance. And that would result from the integration of a synaptic current. And you have an increase if the coefficient G8 is positive. Okay? And this kernel K is a synaptic integration kernel. It has some characteristic time, which is synaptic integration time, which is actually we will assume to be very small compared to all the other times at play here. Okay, so now once this
Is that physiologically reasonable?
It depends on the cases, but I think so. Yes, for what we are doing, we are studying. Yes, but you'll see. I mean, we don't need to do that actually in the model. But if we do this assumption that it's very small compared to all the other time scales, then we can solve the problem very efficiently. I will tell you. So once this current has entered the cell, so the potential will increase, then it will relax before another input arrives here due to the leakage term here. And you have the characteristic time, which is G over C, C over G, sorry, which tells you how fast this potential will relax to the rest value. Okay? So we have inputs here, which can be either positive or negative depending on the sign of the synaptic coefficient here, and you have relaxation. And then there will be inputs also coming from all the other cells which are not measured, so plenty of them. And we just model that through a stochastic term here, the Gaussian distribution.
How big are the relative coefficients of the last two terms?
This one and this one? Okay, so it's a very good question. So it depends very much on the... So for instance, for retinal data, so we had some data by Michael Burr in Princeton, where he showed a movie, The Salamander. So it's a short movie about 26 seconds, and it was repeated 120 times. So you have a lot of repetition. So you can see how much the activity fluctuates from one repetition to the other one. That tells you something about the influence of the stochastic term. And it's very small. So it's highly reproducible. Now, it's true also that this kind of reproducibility result depends very much on the kind of stimulus you are presenting. If you have a highly non-structured stimulus, it's a more random time, then the result may be less reproducible. And there were papers by Marcus Meister, for instance, on the retinal activity too. So in the case also I will talk about, we assume that this term is small compared to this one. Okay, so that's the dynamical equation, okay, for the leaky integrated fibers.
So that may not be true in cortex.
No, that may not be true. But again, that's one of the assumptions we have to do in order to get some efficient way of solving the problem. Okay, so the basic two assumptions are this one.
So you're going to assume that the noise is small at one point?
I'm going to assume that the variance of the noise is small. That doesn't mean that the noise is small. So, it's like saying that, okay, I will tell you exactly what it means, just in one second. That means that you can have large noise, but it's very unlikely, okay? And actually we'll have this kind of evidence later. Okay, so this is so very well known, leaky integrated fiber equation. And then if the potential at some point crosses some threshold, and that happens here, then the potential is reset to the rest value. It can be reset immediately to the rest value. After some time, there might be a refractory period. There are plenty of different details that you can put it here. Okay? So for the black pattern, you get and each time the potential is crossing the threshold, you get an emission of a spike. Okay? So you get one spike emitted here in the black pattern, and for the gray input pattern, you don't get anything. Okay, so the neuron remains silent. So this is the model we are looking at. So again, this is a problem. You are given a raster plot here, and you are asking what is the, for instance, I look at the activity of neuron number 5, so you get a set of dots. I don't know if you can see anything on the screen. But here in this small time window, you have 3 dots corresponding to 3 different spikes emitted by neuron number 5, and you are asking what are the connections from all the other cells onto neuron 5. Okay. So that's the question. So what do we know about these connections? Actually, we don't know the values. But we know the following thing. We know that the internal dynamical variable of neuron 5 is something, so potential D, and we know the times at which it crosses the threshold, each time a spike is emitted. And we know also that right after having crossed the threshold, it's reset to a 0 value, basically, a rest value. Okay? So we don't know anything about what happens in between, but we know the extremities. We know the values of a potential at the extremities of the interspike intervals. Now, in between, assume that we had some idea about the synaptic coefficients, some guess values, okay? Then each time so for instance here, you get a spike coming from this purple neuron, number 4 maybe. So which has a positive synaptic coefficient from 4 to 5, okay? And in that case, you know that the potential should increase. And that will be, as we assume that the connections that do not change with time, then you get another increase, a similar increase, the next time a spike is coming from this purple neuron. Okay? The increase won't be exactly the same because obviously the dynamics is complicated and depends on the value of the potential here through the leading strand. But that's the idea. Okay? So now, if we want to have the likelihood of the data given the interactions, you can see that actually the data are just the crossing times of a potential. Okay? So that means that the likelihood of a crossing time given the interactions is just a product of first passage time probabilities. Okay? That's the time at which the potential is passing through the threshold for the first time. Okay? So basically what we want to do is now, if we were able to compute this first passage time probabilities here, that we would like to put the real data, the measurements here, and maximize this probability over the interaction. Okay? So the problem here is that it's very difficult to compute these first passage time probabilities. And why is it so difficult? Because they depend on all the times in between, all the input spikes in between. So a way to say that is each time basically a spike is entering, you are in some way moving the boundary here, the threshold here up or down depending on the sign of the coefficient. So you basically have a first passage time problem where the boundary is moving. Okay? So it's a highly non-trivial problem from mathematical point of view. So some approaches, I mean this problem was recognized a long time ago, and Jan Paninsky, for instance, at Columbia, has proposed some techniques in order to compute this quantity here numerically. You can write down some kind of Fokker equation for this probability, solve it numerically and so on. But you see, this kind of approach is very hard to implement because we have a problem here with hundreds of cells and millions of spikes, hundreds of thousands of spikes. So it will be extremely slow, and there is no way you can actually do that. Okay?
So sorry, can you repeat again, why is this equivalent to a problem with a moving threshold?
Okay, so you can just interpret it as, okay, so the normal way would be fixed threshold, and each time an input spike arises here, then you have an increase or decrease in the potential. Okay, but it's like saying that. Okay, that's it. But I can say it if you look at mathematical formulation of the problem. It's an input spike basically tells you whether the threshold is coming closer to your point or is going further away. You know, if you have, you can say it. I mean, it's just a different way of seeing it, of seeing the same problem. So basically, the fact that you have inputs here. So for instance, if you had a single time-independent current, input current, for instance, then computing this probability would be easy. People have done that for a long time. You know?
Or if we knew all the inputs, then we would know what the first partial time probability is.
Or if you know all the inputs, okay, assume that you know all the interactions, okay? Even knowing all the interactions here makes this calculation difficult. That was my point. assume that you have a guess value for the interactions. Then computing this quantity is difficult. You have to do some numerical equation. It takes a long time. This kind of approach doesn't scale up with the size of
How
problem.
many cells can you do that with?
Okay, so in this paper here, he had one cell, and he had interaction with the previous spikes, some kind of time memory effect,
I see.
and a small number of spikes.
So I guess it would be easy only in the diffusion approximation, where you assume that you have some meaningful coming into the cell. But this is not.
Yeah, but this is not what you want to do here. You want really to have an idea of the real effective connectivity. But certainly you want to have an idea about heterogeneities in the interaction. Yes, you want to do that. Yes.
So, do your assumptions require that you have like an integrating resetting cell, or so you wouldn't be able to do this with like a Hodgkin-Huxley neuron, correct?
Yes, you mean I'm putting back the potential to a rest value right after spike.
Right.
That's correct.
So could you have a more complicated model that is still resetting and still use this sort of analysis, or no?
You mean, can I do the kind of analysis I will show later with the Hodgkin-Huxley cell?
Or a cell that's not Hodgkin-Huxley that resets but has slightly more dynamic kinetics in terms of how it responds to the inputs.
For instance, a nonlinear leakage current here.
Right there.
Okay, so I will answer your question right after. You see why I need this linear model. It's not a fundamental problem. Okay, I could have nonlinear function here, but you will see where I use that. Okay.
Can you measure instead the full memory potential instead of just the spikes, or is it impossible?
If you have
It's a different word.
Yeah.
So this kind of multiple
Yeah.
simultaneous growth, you can't, right?
You mean having access to the intracellular
The
potential?
intracellular-
Yes,
Yeah.
you can. Of course you can. It's just a completely different set of experiments and a completely
Yeah.
different set of
But
data.
far
Can you do
fewer
that?
cells.
But is it possible?
It's far fewer cells.
Okay, so
Yeah.
if you want like a large number of cells, you
Yeah.
mean
Yeah.
you can record just the spikes.
Yeah.
Yes.
I'm sorry, I came in late. Did you explain how you do this measurement for a large number of cells?
Well, I'm not doing it myself, but
How it's done?
but I just said very briefly at the beginning that people use material for the race. That's just a picture of the material for the race here, which is just put under the retina, for instance, but it could be in cortical system where people use electrodes here. That's just a picture of the electrodes, the shanks, which are just inserted inside the cortex.
And touch many cells.
Okay, so it depends. So some electrodes will be just by chance very close to 3, 4, 5 cells, okay, and we'll get a measure of the extracellular potential, which is a linear superposition of the different extracellular potential of the cells. So it's very hard to disentangle.
Right.
Okay, so that's what spike sorting is about. Okay? Now other electrons won't pick up anything. Okay, so it depends very much on the density of electrons compared to the density of cells. So
Actually, even this data that you have is a bit of an idealization that you have access to. The spike training for 100 cells.
no, we do have the data.
You can
That's
actually
it, yeah.
extract.
Yeah.
This data. But
This
is
data was
showing
given to us
real
a
data.
year ago, 2 years ago. People do have this data. Now, obviously, there might be some mistakes in the spike sorting approach. Okay, so we shouldn't think that these data are perfect. Actually, you can see that. I mean, people do it in it takes a lot of time. It's very difficult to do. But sometimes you can see that there are two spikes which are very close to each other, emitted by the same cell. That's not possible. There should be some refractory variance. Okay, so there might be some mistakes. I think a mistake sometimes is that it's very difficult to. People may think that, for instance, you have a train of spikes that it's emitted by a single cell, while there might be two cells, for instance. So you would like to assign which spike is coming from cell 1 or to cell 2. So this kind of mistake can exist.
So they're reduced to a minimum. I mean, it's still electric, so it
Yes.
knows what it is.
And it's certainly improving. Very good.
So that's
Very good people.
a separate art.
Yes, I mean, I'm not saying anything about that. I'm just assuming that these data, are given to me. I mean, it's true. They are given. Okay, so I say that this numerical approach does not scale up with the cycling problem. It's just impossible. So what we are doing here, the approach I'm proposing is something which is simpler, in a way, and which scales up with the size. So it's based on let's say, a small variance of noise approximation. Okay, small noise approximation. So what does it mean here? I'm assuming that the variance of a noise here of the stochastic term is small compared to what comes from the other term here, which means that among all the possible paths which go from this point here to this point here, then one of them will have a much higher probability than all the other ones. You know, you have a set of passes here for potential, set of potential trajectories. Actually, since the problem is stochastic, any trajectory is possible, but not with the same probability. So some trajectories will have a high probability, other ones will have a very low probability. Now, if I am assuming that sigma is very small, then that means that one of the trajectory will have a much higher probability than all the other ones. So that's something that
What if you have like two paths which similar weight, but different, completely different path?
well, it's very unlikely that you will get something like that, okay?
How do you know?
Okay, because I can solve, I can find actually the classical path, okay? So what I can find
I'm
it.
confused. If you have
Yeah.
two neighboring cells with similar connectivity and they fire, or if you have one with a stronger connectivity, it's hard to. Naively, one thinks one could cook up things with very similar properties.
Okay, so let me say again. Assume first that you know the interactions. So I'm looking at the direct problem.
So you're
And then I'm
fixing
trying, I will solve the inverse problem later. I'm fixing the interaction, so I'm computing the likelihood here. Assume that you have a guess for the interaction, you are fixing the interaction. Then the problem is well defined and you're looking for a distribution of paths here, or a potential. Okay? What I'm saying is that in the small sigma case, where sigma is limited, where sigma is the variance of the noise, one path will dominate. And it's very similar to what people do in physics, for instance, where you have a particle trapped in a well, for instance. And eventually, and you have a barrier here, and eventually the particle will escape from the well and will go to a deeper well, okay? If you are really at 0 temperature, then the particle will be stuck in this well here, in this high well, for an infinite time. Now,
that one mechanical.
okay, classical path. Now, if you have a small temperature, but different, very small but different from 0, very small compared to the height of the barrier here, then you have a lot of time to spend in this well. At some point, there will be barrier crossing, okay? And then there will be relaxation in the other one. And the path, which is the reaction path, like in chemistry, if the temperature is very small, it's highly deterministic. Other paths have a very low probability, okay? And this is what is going on here.
Although intuitively it's not clear. That's going to be the case.
Well, okay. So I can tell you how we find the path. Maybe that will
Well, if you have the weights and you have very little noise, the system is deterministic.
yes.
So why is
Well,
it?
no, you still have, it's not deterministic because you don't know when your cells fire and which cells fire.
No, I'm
No.
You could be in the chaotic region.
I don't
it's
understand.
nothing, so it's,
How do you know when the cells fire and which cells are firing?
no, wait.
Yes.
No, wait.
You're looking at a specific cell and the connected
Wait.
cells. Okay,
I'm supposed I know, so the interactions are fixed.
right.
Okay, a guess, fixed. Now I want to compute this quantity, and I'm saying numerically it's very hard. I will look for an approximation of this quantity in the small noise limit. So I'm given.
I'm confused by smart. I mean, is there any input? Is there a non-noisy fixed input? That's what's confusing.
Oh, okay, sorry. You mean in this term here?
No. The next term, the K.
Yes. So here's the point. I'm giving the interaction here. I'm giving the times of the
Oh,
spikes.
you're given the time.
Yeah, I want to compute this probability. So as
Okay.
and then I want to compute. Okay, so you are given this. You are given this. You want to compute this number. It's a number.
Fine. Sure.
Okay, that's. I'm sorry, I was not clear about that.
So for given, okay.
Yes, it's a number. Okay, I want to compute this number. I said it's very complicated, but I am looking for an approximation, something which can be computed very fast.
sorry, I think it depends on the form of the equation. If it's highly nonlinear, you can have a very small deviation from the initial value. And
It's linear.
I mean, it's highly nonlinear because you are here. The nonlinearity is here. Okay, it's a linear equation of potential, but if you look at the different potential of the other cells, okay, they will enter in a very highly nonlinear way in this equation, okay, through this threshold mechanism.
So my question is, how do you make sure that even the deterministic limit you have, I mean, even deterministic limit you can have like small deviation from the initial value, but the path could be quite different.
Yes.
So the small
I agree.
noise limit does not guarantee you have just one path.
No, that's a different thing. You are saying that if I change a little bit the time, for instance, spiking time, for instance, I will have a completely different path.
Yes.
I agree. That doesn't mean that there is not a single path with a maximum.
So now,
Probability.
what do you mean by path?
So a path is a trajectory of a potential. Okay?
No, I'm being very stupid. You're fixing J,
Yes.
you're fixing T.
Yes.
Then the potential is fixed.
No.
Why not?
Potential is obeying this equation. This is a noise. It's a stochastic evolution
But you
equation.
set the noise to zero.
No, I'm not
In
setting
the
the noise.
deterministic case.
No, I'm not setting the noise to zero. I'm
Okay.
I'm saying that the variance of a noise is very small. Which is not saying that the noise is small. Okay, that's the question you asked before, okay? You know, it's like, think again about this analogy in physics, which is simple in a way. You have this particle here. If the noise is equal to zero temperature, you stay here. No quantum effect, you stay here.
Right.
Now I allow for very small temperature. The particle will move here, and sometimes it will have larger excursions, okay? Larger fluctuations. And then it will decay again and so on. At some point, with if you wait long enough, you will find this, okay? The path here, so the noise is very small, but you will have very large excursion. It's a large deviation.
Sure.
It's very unlikely, but they can exist. So that's the idea of the approximation here.
So, sorry, maybe an analogy would be that if you fix the spike times and the weights, right, and the noise is zero, then you would have, probabilistically, you would have a delta function.
Yes, only a
And now,
few possibilities.
if you turn on a little bit of noise, then heuristically you could imagine you know this delta function
Exactly.
opens up, but
Yes.
then it clearly tells you that there's a dominant path. The path
Yes.
where the probability is much higher than any other path. And this is
Yes.
that's what's happening.
Yes. So this is how we compute this path.
Then if you fiddle around with the spike times, then you may end up with the problems that he was mentioning, that if it's a non-linear system, then you might end up into a chaotic behavior, even though there's no...
No, I mean, if I understood you correctly, you're saying that if you change, for instance, a little bit the spiking time, then you have a very different path.
Yes, yes.
I agree. Okay, so here is the problem. So in this small sigma case, I mean setting, I want to find the most likely path, given the initial condition, which is right after spike potential is zero, and right before spike potential is the threshold value. And there will be a path in between. Now, what are the equations for this optimal path? What we call classical path in physics. So there are two possibilities. Either we are in the blue regime, which is in the sub-threshold regime, or we are in the purple regime. So let me start with the blue regime here. So in the blue regime, actually the potential here is, this optimal potential is obeying exactly the same equation as the stochastic equation I mentioned before. It's exactly the same equation. With a huge difference is that this term here is not a noise anymore. It's not a stochastic noise anymore. It's not a stochastic one. It's a deterministic quantity, which is solution of this equation, which is a motion, which is, okay, so you take just the first part here with the plus sign. So it's well known in physics, that's called the motion in the inverted potential. Okay, that's instantaneous path and so on. But you can show that. So how do you want to, this was shown actually by Paninsky and other people, it's easy to show that the optimal path is solution of this equation. And how do you do that? Actually, it's easy. What you're saying that if I take the first part here, the last member, I just subtract this part here, I get that the noise is something, which is a function of potential. And the probability of this potential will simply be in exponential minus 1/2, let's say, the integral of the square of the noise over time. Okay? And you want to minimize this integral of the square of the noise. You get a differential equation for potential that you can rewrite in this term. So the interpretation of this deterministic noise, I don't know how to call it, it's an input current.
I actually don't understand why do you put a deterministic equation here? It's noise, right? I mean, when you put the noise, the Gaussian noise in it, you have the path integral. this is a noise term. Why do you have a deterministic term?
I'm looking for a most likely path, the one with largest probability. It's not a stochastic path anymore. It's a deterministic one.
Oh, so the equation, the second equation is essentially the class equation. Basically, you
Yes.
get from this minus a the square, this Gaussian.
Yes, so you can say that the whole information about this
Yeah,
is
this
here.
is the result of the fact.
Yes, exactly.
Okay.
Okay, so the potential is obeying the same equation as before, but this term here now is not a noise anymore. It's a deterministic input current, okay, which is given solution of this equation. Okay, so now we are coming back to your question. You know, this equation, if I'm giving the times here, and we have this linear equation, I can solve it very easily. You get an exponential here, you put it back here, you get a simple solution analytically. If you had a nonlinear equation and a nonlinear leakage term, then that would be more complicated, okay? But you could have a numerical approximation for the solution.
How much more complicated? Because you could approximate a very large number of cells if that's quadratic or cubic, very.
Yes, so you could, I mean, don't have exact analytical expression, which I like, but I mean, that could be done. I don't think it's a major difficulty. While the assumption that the variance of the noise is small is a major assumption here, and if this assumption is not correct, everything, I mean, I cannot use this kind of approach, okay? Well, here it's not a major assumption. It just makes life a little bit simpler.
This maybe, if I differentiate, what are the, how can I think of, what's the derivative of k with respect to t?
Okay, so k is, we don't have, so k is something which is causal, okay? So it's 0 when the argument here is negative, and then it will have a bump over some, let's say, synaptic integration time. And the integral is 1, let's say, like, just to give the scale of the integration. So it's something like that.
Like a double exponential.
Yes, exactly.
Double exponential?
Subtraction of two exponential with two different time scales. Horizon. So that's one possibility. But now, the other possibility is actually that the potential will be very close to the threshold level, on the threshold level, without crossing it. It's possible if the variance of the noise goes to 0. Otherwise, if you have a finite noise, obviously, if you get very close to potential, at some point there will be a crossing, and you will get a spike before the end of the spike, interspike interval, which is not allowed. Okay? But in this setting of very small variance of noise, that's possible. So in that case, the equation is very simple too, because we know that the potential should be equal to the threshold value, and the derivative of the potential should be equal to 0, so that gives the value of this deterministic noise, if you want, input current. So we have two sets of equations, this one and this one. Both of them are simple. What is hard here is that we don't know when we should use one or the other one. We don't know where we are in this blue regime or this regime and so on. Okay, so the problem is you are given the initial condition and the final condition here, and you have to find the succession of different regimes such that you get the optimal path, the path with the maximal probability.
So what you really mean there's B equals B threshold minus X? Is that right?
Yes. But if you do sigma going to 0, mathematically it gives this. But in practice it means minus, yeah.
Just
but I'll come back a little bit. Okay, so
it's an extremization problem of piecewise linear. Well, piecewise, not linear, piecewise solutions.
yes, yes. Yeah, well, yes, exactly. But the problem is that you don't know where you have to put the answer to intervals
So
here.
in a completely different context, these kinds of problems have been considered by my thesis advisor. These kinds of extremization problems. I can't, off the top of my head, I can't remember the reference, but if you look at his work in the late 70s, early 80s, you can find some, from just purely applied mathematics point of view, these kinds of extremization. He applied it in particle physics.
Yeah, I mean, I'm sure there are plenty, you know, in control theory or so, there are plenty of, I mean, there is a huge literature about this kind of problem. I was not aware of this particular kind of problem exactly here, but I don't know. Okay, so actually this problem of finding where we have to go from the blue to the purple and so on is something which is not easy to solve. But now comes the second assumption. We're assuming that this integration kernel is very close to a delta function. So that the time scale of integration of inputs is small compared to all the other time scales here. Okay. Now, if we have this second assumption, then actually we can solve this problem in polynomial time. So what we can show is the following thing, is that this purple regime, so the contact regime, might be of two different kinds. So either you have an isolated contact, that is that the contact actually of the potential with the threshold here is just an isolated point. It's not a whole interval. Okay, so the potential just touches the threshold and then goes down without crossing the threshold. And that may take place only when there is an input spike. So at a time corresponding to an input spike. That cannot take place at a different time in between two input spikes. This can be shown. And if you have such an, what we call active contact, then the value of this deterministic noise will increase. The value of the noise after the contact will be larger than the value of the noise before the contact. So I don't want to show all the mathematical details, but just believe me that you can show that. So it's a very simple rule. Or, second possibility, you get a contact in between two input spikes, okay? So you have two input spikes here, two consecutive input spikes, and you'll get a contact here during which the value of the noise is constant. But these are the two possibilities. Okay? So using these only two possibilities, it's actually possible to solve exactly the problem, and I want to show you some examples. So here's an example of one cell, so the black cell, receiving inputs from the green and the red cell. So the green synaptic coefficient is negative, and the red one is positive. Okay, so you are given this set of spikes here. Okay. And you are given also the synaptic coefficient, so it's always a direct problem. So how do you find the optimal path? Okay, so it's easy. So we start with the first fact here, and we say, okay, so here is the value of potential right after the emission of the spike, which is 0, and I say it's 1 right before here. 1 is the threshold value. Okay. So one thing you can do is compute, say, okay, suppose there is no active contact, so I will just say there is only the blue regime to go from this point to this one. So I can solve this equation and find the coefficient here, the integration coefficient here, in order to arrive exactly at this threshold value at the end of the interspike interval. It's very easy to do, and you get this black trajectory for potential. That's the optimal. Or you could say, well, maybe I could have actually crossed the, I could have active contacts at times which correspond to the input spike here, and if I got a contact here when I get the green input, I get this trajectory here, or I get the red one here. Now, obviously, neither the green nor the red are correct because they cross the potential here. Okay? So the black is the good one. So it's a very simple case. Nothing happens very interesting here.
You mean 1 is the threshold?
Yeah, 1 is the threshold here. So what you get is the optimal path of the potential here, and you get the optimal value of the noise here. Determines the input term. Okay? Now, what happens during the second interval? That's more interesting. So I could do exactly the same thing. Here is the second interval. Okay? And I say, okay, look only at the black and the two green curves first. I could do exactly the same thing. Suppose that, okay, so I start from this condition, I want to reach this one, and I suppose that there is only this blue regime in between, okay? And I get this black trajectory here, the potential, which is certainly not a good one because it crosses the potential at intermediate time, so it should be rejected. Or I could say, well, maybe I've got an active contact, so isolated point contact before, and that may take place only at this point or this time here. I get two other trajectories, which are the green ones. So you can see this one is contact here, and this one is in contact, the second one is in contact here, okay? But none of the two here are acceptable true, okay? Because they cross the potential.
The threshold.
Yeah, they cross the threshold. So they should be rejected. So what happens here is that we are actually in the second possibility I mentioned here. We have what we call a passive contact. That is a contact during a finite time interval here, during which the noise is constant. So we have a noise like that, some passive contact here for some time, and then the trajectory has to restart, to resume, to reach the threshold value here. So how can this be done? Well, if we just look at this blue trajectory, you see that it would go to a value of potential which is too low. That means that the noise has to increase in order to boost the potential, okay? And in order to do that, we need an active contact in between. So when can it happen? Only when there is an input spike here. So one possibility is an active contact exactly here at the first time here. And if you do that, and then you get the red curve here, okay? So the red curve is good here in between, but still the value of the potential is too small here. That means that you need another active contact here in order to reach this value here. Okay, so I just do it by moving my hands, but this can be done by solving a set of equations, easy equations. And you get this optimal shape of the noise here, and this optimal path for the potential. So if you know the currents and the interaction, you can solve all of these equations and find the best path in a time which is like the square of the number of cells times the number of spikes. Okay? So the number of spikes usually is proportional to the number of cells too. So the number of spikes is usually the number of cells times the duration of a recording times the average firing rate. So it's basically cubic in the number of cells times the duration of the.
So you do this numerically?
Yes, but you have a set of equations to be solved, and this equation can be solved analytically.
Right. So the elementary time step with your numerical...
No, that's the point. I don't have elementary time steps. I don't have it. I don't have to discretize over
Not the
time.
number of spikes. Then.
Yes, otherwise you would have, elementary time step, okay, you will have a factor which is basically the intertime divided, and that's 100, 1000.
Yes, exactly.
So this is what we gain here.
Going from active to passive, you don't need a spike. It's only when you go from passive to active that you need a spike.
So here it's a particular case where the active contact is right at the end of a passive contact. But that might not be the case. Sometimes you get potential, passive contact, and it goes down, and then you get an active contact after. You can have an interval in between. It's a special case here. Okay? It might not be the most pedagogical illustration. Sorry about it.
I'm confused. If you have a passive, then what triggers it starting to decay?
Okay, so you could stay for hours. So the point is, okay, so you start here, you have it in the passive, okay. Now you have to determine the length, the duration of this passive contact. Okay. So the point is that you need to reach the potential, the
I
threshold
understand, but
potential.
what triggers it? What tells? There's no, you can switch from passive to active at any time. I thought there was some correlation with the.
No. I mean, I have to find the optimal duration of the passive contact in such a way that I will reach this threshold at a later time. So I have to arrange active contacts in between. Sometimes I don't need, and I need them. Usually I need them at least one of the active contacts. So I have to tune the duration here in order to find the right active contacts later at later times in order to reach this threshold here. But again, so it's a subtle optimization problem, but that can be solved in very little time.
Do you specify the end time when you do the optimal path?
Here,
Yeah.
I do, yeah.
But the optimal path depends on the ending time too.
Absolutely.
And so how are you going to vary the ending time?
No. Again, I want to compute this quantity. So here I'm assuming I know the interactions. I want to compute this quantity. So give a set of spiking times. I want to compute this quantity. And
so the end time is specified.
yes, absolutely.
Now, is that a variational problem?
It's a variational problem. It's
You
an
have to find
optimization problem.
The curve that satisfies those two sets of equations in the way that you get.
Yes.
But here's my question: how do you know the end time? I mean, you said you fix the time, but don't you have to vary it at the end, but with this ending time?
No, I mean they are given by the experiments.
No, but you want to maximize at the end, right?
But I want to maximize over the J's, not about the experimental data.
No, not the peak time. What I mean is when you specify the path, it's not what happens in between. It's the beginning and the end time. This end time.
You know the beginning and the end.
Yeah. So this time interval.
Yes.
You fix it.
Yes, because you know the spiking times. They are coming from the data.
I thought this I end is something in between, the pulse in between. Right? Means, but but this one is not right here. It's
So the beginning and the last, they are the spikes of the neuron I'm considering. So I'm considering each time interspike interval, one after the other.
Oh,
Yes, each of them. Then I know the first time and the last one, okay? And then in between, I've got times corresponding to the inputs. And they are known from the data too.
Yeah. So, and this interval is also known.
It's known. Then once I've solved and found the optimal trajectory, I move to the next time interval and so on. I do all of them. So that's why it's proportional to the duration.
So in the end, you are varying the j at the end. That's what you want to find.
Yes, so
Okay, all right.
here, everything I'm saying here is that it's for fixed j. But now, if you are able to compute this path and the probability of the path, then you are able to maximize the j's.
Yes.
Okay? So we fix the j, find the path,
Yeah, but still
and then
keep the
yes, but the time they never move, they are the experimental times. So we find it, and then we optimize the j. It turns out that it's a convex function of the j's. It's like this. So that can be solved, that can be optimized easily. So we do gradient descent. Actually, we do some kind of Newton-Raphson technique.
So the input times are given because, what is it, the spikes from the other neurons they recorded from?
Yes, they all come from the data. Yes.
Right. But then of course you don't know whether these neurons are connected to them.
No, I'm not assuming anything. I could assume something in the prior information, but I'm not assuming anything. So maybe at the end of the process, I will do a lot of calculation and I will find that the connection is zero.
So this V is for a particular one, right? The VI. Right?
Yes.
So you have lots of.
Yes. So you know.
You keep the same. Yeah. Okay.
Exactly. So that was one neuron and one interspike interval. Just repeat it for the whole duration and then do it in parallel for all neurons. But it's a parallel process, so it can be parallelized very easily.
So you can, this is coupled the equation.
They are uncoupled. They are uncoupled for the following reason, because they assume here
Yeah.
that the noise here, noises are independent from cell to cell.
Mm-hmm.
You may think it's not a good approximation or a good assumption. So, you know, if you go back to the original data, you may think actually if you have an input, a structural input stimulus, then maybe there should be some correlation in noise, you know, from cell to cell.
But the JI gives you the correlation of this particular cell with the other cell.
Yeah, so you could have two kinds of correlation. You could have correlation coming from the interaction here, but you could have correlation coming from the stochastic terms as well.
Sure.
So I'm assuming that there is no such thing. If you have no correlation from this, then there are just independent problems you have to solve. If they are correlated together, then you should solve all the path, the optimal path together, which is a harder problem.
No, I think that this J coupling already coupled the different V's, right?
Yes.
So it's not independent equation, it's a VI. So it's a vector you're solving.
No, I agree, but what I'm saying is that this is true. You have a product here of first passage time probabilities. It turns out that the inverse problem, when you do the inverse problem, they are independent.
So what's the intuition for that?
Now we can talk about that, but I'm very sure about that.
Mm.
And this comes from the assumption that the n eta I, you know, I mean there is a missing index here. That's true for one cell. Okay, that's the equation for one cell. So eta sub I for cell number I. I'm assuming that eta I is independent. From eta j.
That's fine, but what I'm saying is the coming in, the other deterministic part depends on the other V, the other potential of the cells.
Yes, through the inputs here, but only through that. You know, the equation for potential, then the potential for cell I adds its own life, you know. It's driven by its own current, and that's all.
Calculating the spike train from the V's, it's calculating the V's from the spike train. The
Yes.
spike train is fixed.
A
Yes.
spike train is
Yes,
the input.
actually
it's the
the-
input.
yes.
It's not like the feedback of the
No.
No, not, you know,
Okay.
no. So from this point of view, it turns out that the inverse problem is easier.
No, okay.
Because you just fix it once more, the data, nothing else. Okay, so let me, just a few seconds. So actually, everything I said here was in the sigma going to zero limit, okay? If sigma is not equal to zero, then you will see some deviation. That means, for instance, here I give an example. We have run some simulation, for instance, for the optimal. So what you find, so you are given some spikes here, the tiny given, and you solve optimal path, find the optimal path, and you get the black curve. Okay? Now, what we do, we just run a very simple integrate and fire simulation, okay? And compute the average value of the potential over a certain number of samples. And if sigma is very small, we get a perfect match with the optimal solution. If sigma is not small, and here are some values, but whatever, you get that the potential is pushed down with a very simple effect that it has to be further away from the threshold, otherwise you would have spikes at intermediate intervals. That's an obvious effect, okay? So it's very difficult to solve a non-zero sigma case, but you could do that in some very empirical way. Just saying that, okay, let's try to say that actually there is some kind of effective threshold, let's say, which is not one constant, but which is something which depends on the value of sigma. The larger sigma, the further down you want to go with this effective threshold. You want to push the optimal path down. And we have some way of doing that, but again, it's very empirical. What I've said before, I think, is mathematically correct in the sigma going to zero. Sigma different from zero, but we have some modification of the algorithm I want to mention too. Okay, so what can we do with this kind of thing? So you get optimal values for the interactions. Typically what you get is, once you have solved this optimization problem, you get the value of the interaction. So here are these are coming from the analysis of 40 different cells. Okay? And I consider one cell, so cell number 1, whatever, and the interaction coming from a cell number from 3 other cells. Okay? And what we can plot is the posterior probability of the interaction. So when we solve the optimization problem, we find the most likely value. So here is, that I choose a pair of cells with a negative value, a pair of cells where the value is essentially 0, another pair where the value is positive. Okay. So you get this curve, and actually what are the error bars in these estimates, which is important. You don't want to know only the value of interaction. You want to know the value of interaction plus minus some error bar. Okay? And this error bar will decrease. That means that this curve here will get, the curvature will get bigger and bigger if the number of data is bigger and bigger. So you get something more and more concentrated if the recording is very large. So you have an idea about that. So in particular, if you get an interaction which the most likely value of the interaction is non-zero, but the error bar is so huge that actually it's compatible with 0, just forget about the interaction. So it's important to know what is the size, the width of this curve. So we have tested on different synthetic data, and I think it's fast. To my knowledge, it's the fastest technique for integrate and fire. To solve the inverse integrate and fire problem, with, again, two major assumptions. Small sigma and integration kernel is immediate. Okay? Small integration time. Now, there are other techniques which have been developed, and one actually, I think the most recent and powerful one was developed by Koyama and Paninsky 2 years ago. And what they do, so one of the assumptions they use is the same as ours, which means that it's only the small sigma regime. They look for the optimal path, but they do it by time discretization, and they solve the equations. Time, they discretize time in time beings which are much smaller than the interspike interval, and they solve this thing. So I think it's much slower. But the good thing about their approach is that they don't have to assume that the integration kernel is instantaneous. Okay, so
Can you put the synaptic delay?
okay, synaptic delays can be introduced here, also in our approach. I don't think it's a problem, but the only thing I can put a synaptic delay. But the point is that I need a delta integration, but I can shift it, each time. That's not a problem. Here I'm talking about something different. They can assume that this K kernel could be something, can be anything. So their technique is more general, but I think it's much slower in this particular.
So
there is a trade-off,
So your kernel K was a delta function.
It's a delta function.
Can they also use a different model of neurons? So for example, the nonlinear equation, because if they're discretizing time anyway,
oh yes,
they have to integrate the equation, so they're not bound to use the model that you can solve analytically.
Yes, that's true. That's true. But again, what we discussed before. If we can solve it analytically, but we have a very good approximation scheme with a few parameters, I think we can handle that too.
So have you applied this to real data?
Yeah. So I just want to show preliminary analysis. So real data, so retinal data, and we have also some analysis of cortical data, and I'll talk about that later. So I think there are important questions to be answered. So we look at a set of cells, so a few tens of cells here, which were given to us by Marcus Meiser and Harvard. And so the first question we ask is, okay, so you can run this algorithm and you get a bunch of interactions. So what do they mean? Well, first of all, before talking about what they could mean, actually, is the problem well behaved? Okay? So it's an important problem because the retina is that big. Well, okay, it's much smaller, but anyway, what you record on the retina is very small compared to the retina. Okay, so that means that you record a few tens of cells among thousands of them. So what does it mean? That maybe, for instance, if I come with an electrode array, I get some interaction, and then maybe a few years later, electrode arrays will be denser and they will be bigger. And then maybe I'll get a completely different set of interactions, assuming that we can use the same standard retina. Obviously, that's not the case. But the question is, how much what you infer depends on what you can't measure. So it's an important problem, because you want the problem to be well behaved. Okay? So it's a question which can be answered to some extent in the following way. So here, so we look at spontaneous activity recordings. So here, the visual map. So each dot corresponds to the center of the receptive fields of one of these 32 cells in the XY plane. And we were able to infer some interaction. I will show you a map of interaction in a later slide. And now we ask the following question. Suppose I consider a pair of cells, so it's a 9, 8. I look at the interaction between going from 9 to 8. And what I do is that, so I got 32 cells, I look at these 2 cells here, so I can remove from the dataset one of the other cells, okay? And then compute the new interaction between 9 and 8. And I see whether it changes or it doesn't change. So what you get here is the histogram of changes when you remove one of the cells. Okay? That tells you how much you have collected, I mean, if you have network-mediated effects or not. And you see that you get basically no change for all cells except cell number 12. And cell number 12 is here. Well, I mean, the receptive field center is here in the vision. Okay? It's not very far away. Now you can do, for other pairs, you find basically no effect for these 2 cells here, and you get strong effects for the pairs 5 to 1. And 5 to 1 is here, and you see that the cells which are important, which can change the interaction here, are located in the neighborhood of this pair. So
this cell is important because this is information that comes from you having solved the inverse problem.
Yes. So, but what we never get is, you know, remove this cell and get a change of interaction here. That means that the interactions do depend on the activity of the other cells, contrary to cross-correlograms, where you can remove something and that doesn't change your cross-correlogram, okay, obviously.
What is it doing? I'm sorry.
But it doesn't change, but there is no consequence if you are looking at the activity of a cell which is far away. Far away meaning 400 or 500 micrometers.
So in this case, you can solve well problems where the connectivity is short range. Because in cortex, where you have long range connectivity over millimeters, you would be having problems.
Yes. Because, but anyway, here, so what I'm saying is, you know, it's very limited. What I'm saying here is that, see, we have some problems here because Marcus Meiser recording is quite, it's a little bit old, it's from 7 years ago. And the array he had was not very dense. So that means that he was missing a lot of cells, you know, in between 2 cells, and so his estimate is about, you pick up about 20% of the cells. So what we are saying here is that, anyway, we get some effective interactions, you know, because even if the interaction we get do not depend on what happens very far away, since we are missing 80% of the cells, we certainly don't get the true interaction. So in Michael Berry's experiment, he has something where he believes that he claims that it's really, I mean, it's true, it's really dense, so maybe we pick up all cells. So this kind of problem would disappear.
So could you repeat what the problem would be if you were doing a cross-correlation analysis?
Well, if you do a cross-correlation analysis, you don't have any problem, because you look at the pair of cell I and J, and whether you measure the activity of K or not, that doesn't change the correlation you get between I and J. So it's
do have a problem, in fact.
Well, it depends on what you call a problem. You have a problem because you are looking at a correlation, not an interaction. Okay, so it's an initial problem. Okay, major problem, conceptual problem. But then the technique is very fast, and it's very robust in some way. Now, if you want to solve the inverse problem, it's a global problem, okay? Now, obviously, the interaction between I and J will depend on the activity, okay? But I just want to understand how much it depends, and if there's some kind of distance effect, that the effect will decay with some distance. And for the case of spontaneous activity in the retina, it's a very special case. It seems to decay nicely with distance. That's all I'm saying.
Is this akin to some sort of finite size effect? Like studying finite size effect?
It's more correlation, correlation effect. You have some correlation lengths and.
I mean, one obvious thing you could do to check things is just take different subsets of the data, see if you get the same kind of I assume you've done that.
Oh, but that's basically what we are doing here.
No, I mean subset like temporally.
Oh, okay. Yes, we do. They are completely compatible. You get larger error bars.
Right, but you
Yes.
get the same answer.
Yes. Obviously, if you have a completely different, very time-dependent stimulus, then that could be different here. But here it's just spontaneous activity.
So how does your past probability estimation these connections compare with the cross correlation data? Like how many of them are compatible, how many of them are not compatible? So in this way you can infer some of them are physical, some of them are like just like what you showed at the beginning.
Yeah, so
like the sort of not real there, but it's the correlation.
I understand. So I don't have a map here because I wanted to show you a map, but just to answer your question is usually when we look at the network of strong interaction and the network of strong correlation, what we find, the main thing, the first thing I can show you a paper. The first thing we see is that the network of strong interaction is sparser than the network of strong correlations. Sparser. Sparser. Okay.
Than the
Network of strong correlations. So, okay, now if you want, I want to say it in a better way. It has a nice, okay, so if you want to compare them, so for instance what you can do is, you say, okay, I got n cells, n times n minus 1, the two possible interactions or whatever, and I keep only a fraction, let's say the 5 top percent in an absolute value, can be negative or positive, okay? And then you look at these two networks, so they have the same number of links. So in one case, and I show you, so this is what we get, for instance, for the integrated fire models of spontaneous activity. Okay? And this is what we get with maximum entropy model.
Sorry, what are you plotting there?
Okay, so that's a map of interaction. So down the visual, so that's a visual plane, again the same dots. I'm plotting the strong interactions. So it's a fraction, the top 5% of the interaction. I don't remember 5%, maybe not exactly 5%. In the top interactions, okay.
So the dot is the center of the receptive field of
Yes.
the cell?
Yes. Yeah, I just want to put them in a plane,
Okay.
That's not a true plane of a ganglion cells, but
Right,
it's a plane.
And
And you have a strong interaction. If you get a ball link, that means that the interaction is strong in both ways. If you get a dashed one, it's strong in one way, not in the other way. So integrate on fire, here, I didn't talk about it, I just mentioned very briefly, you can do that. You can do, this is for one model, you can use other models, maximum entropy models. And you get different maps, but they compare. So this is what you get when you use this interaction. If you do the same map, I can show you in the paper, with the correlation, you don't get the same thing. You get one cell, for instance, which is correlated to many other, a kind of hub, and other ones which are completely isolated. And if you put the same number of links, you don't get the same network. You get something much more regular here. But I don't think that's a good way of saying whether this approach has a meaning or not. Okay? What we want to know is whether we can do prediction with that. Okay, so I just want to mention that in the conclusion.
so once you solve your inverse problem, you can actually look at the correlations between the model given. Right?
You can look at the correlations.
So the correlations,
Yeah.
and they presumably look different from the interactions.
Yes, so this is what we were discussing. Or maybe I didn't get your question.
I mean, okay, I guess. You can use it as a predictive tool.
Yeah, so I mean, there is no miracle. I mean, if you get a very strong correlation between two cells, you'll get a strong interaction, obviously. That's not possible. But in some cases that's not true. I mean, you get, for instance, a mild incorrelation and you won't get an interaction because you will find a path going through other cells that explain the same correlation. There is no need to put any interaction.
So really, when you read
So you get something
interactions,
simpler.
Yeah, so direct interaction. Direct interaction.
Within the model.
Functional interactions, so it's a spike causing another spike or something like that, but it's not necessarily a function, right?
Yes.
Physical.
Do you have any understanding, I mean, the two different methods? Fairly different results.
That's not too.
No?
That's not too bad.
I mean, the maximum entropy one doesn't seem to, that doesn't have the option for asymmetric, is that?
No, it does not have. I mean, you can have more complicated maximum entropy models where you can put interaction, asymmetric interaction, but here you get only symmetry. That's a kind of icing model. That was proposed first by 5 years ago by Bialek, Michael Burry. They had a paper.
Could that account for some of the differences?
Certainly, yes. And you have more parameters here in the model. You have a leakage time, for instance, that we don't have here. Or here you have a binning time window. You know, the configuration are discretized over maybe 10 or 20 milliseconds, where you put 1 if you have a spike and 0 if you don't have a spike. So you get a set of binary words. So you get some time, typical time. Here you don't have, I mean, you have a leakage time. But for instance, cross-correlogram, in some cases you may have a peak, but the peak is not centered in 0, it's just shifted by some delay. So if the delay is 50 milliseconds and you are binning in a time window of 20 milliseconds, you will completely lose it. And the integral amplifier will capture that.
So these two maps, what's your conclusion? Maybe you said, do they look similar enough? Do they look different enough? Or?
I think they look, everything looked pretty similar.
Yeah.
Except that, so the interesting point I wanted to mention is that here, this is for spontaneous activity, here that's for another recording on the same retina with random recurring squares. That was done by Marcus Meitler. And you see that the network of interaction you get is actually not the same. This interaction will depend on the stimulus, the affective interaction. I mean, there is a subnetwork of interaction, some kind of, let's say, almost nearest neighbor, let's say, short-range interaction, which look like very similar. But on top of that, we get long-range interactions, which are clearly induced by the stimulus. So the structure of the stimulus is modifying this effective interaction. So we can't get physiological interaction with this kind of approach, okay? I mean, I'm not claiming anything like that. There should be some support of due to the physiological interaction, but clearly there is something else.
So which kind of predictions can you make?
So okay, so everything I said here was very preliminary. So there are a few things. We'd like to know if this model are predictive. So a few tests that we have done, for instance, which were also done by Bialek and Berry, was, for instance, can you reproduce 3-cell or 4-cell correlations? Because you know this, for instance, in the maximum entropy models, because everything is based on 2 cells. And it turns out, even for cortical data, it turns out to be very good, which is surprising. So for instance, we look at the probability that you have K cells firing in the same time window of 10 or 20 milliseconds, for example. Okay? And that's not something you put in your model. Okay? That's something you can compute from the model and you can compare to the data. From this point of view, the model is really good. Okay, so I don't know what it means, but then you can look at three, I can show you, at 3-cell correlations. So you want to measure from the experiments and you want to compute from the maximum entropy model. And you find a good agreement, fair agreement. Okay. So that means that this model has some predictive power. Now we'd like to test them on a much more interesting situation where, for instance, in principle, if you record the activity for some time, then, for instance, in some cortical systems, if these models are interesting and they can tell you something about the system, then they should be able to tell you how the activity is modified if you do some stimulation on one cell, for instance. Stimulate one cell, how the activity should change. So that's one thing we'd like to do. Okay? I mean, there are very clear tests of whether these models have anything interesting or not, which is a mathematical game. Next question. And one thing I'm very interested in, and we are launching some collaboration with people in Amsterdam and in Leuven, is if you do the same thing for rat I was mentioning. Okay, so you can measure the activity before the learning of the task, get some connectivity map. Do that after the learning of the task, and can you see a change in the connectivity? I mean, is that some kind of, you know?
There's also something better that you can do, which is there's behavioral states that you can create, and so you can try to figure out if you can get rid of rats that are engaged in a task versus what is the spontaneous activity and the correlation when they are disengaged, and see if you have any changes in the functional interactions. That would be a good point of this model, because it's the same animal, it's probably about the same time, and the two interactions are actually going on at the same time in the same network.
But is it better? I mean, it's different.
But it's not.
It's more toward what he wants to test, which is a shift in
It's not
functional
bef-
connectivity.
It's not before or after.
It's not before or after something
Yes,
has been learned, it's simultaneous. So you can do it during learning simultaneously and have a before and after of the two states, actually.
I understand.
I mean, you might expect that since the time scale for connections to form.
They don't
So
form.
this is functional connection.
This is functional
Right.
connectivity, so you don't need the anatomical connection, is what he's been talking about for the whole talk. You don't need the
But
anatomical,
the anatomical connections are still interesting. I don't know.
they are to me, but, but,
But if he sees
but
it,
in a function, like in
Yeah.
learning, it's, the connectivity can be the same, but the way the circuit is engaged is not the same. And so that changes the way the functional connectivity operates.
I mean, point is, if you do it before and after learning, you don't know whether your changes are due to
Yeah, you don't know.
anatomical changes, if you want, structural changes, or whether they're just due to the physiological changes.
So you have
Yeah.
the
It will still-
structural
it will still
process that
not
is
be
going
able
on
to
in
listen
the
to
tangle the two.
but-
Sure.
Okay, so last conclusion is that honestly I don't know if these models are interesting or not, but I think now we have fast techniques, fast resolution techniques, which maybe will allow us to answer this question. That's a conclusion.
Well,
Okay, let's thank